So this portion of the video is going to discuss Carlos Chagas and his contribution to microbiology. Carlos Chagas was a Brazilian physician, scientist, and bacteriologist. He was most famous for discovering Chagas disease, also known as American trypanosomiasis. He was alive from 1879 to 1934. He graduated from the Rio de Janeiro Faculty of Medicine in 1903. Afterwards, he dedicated his career to focus on malaria and its specific disease processes. Malaria is an infectious disease transmitted into the blood by mosquitoes involving a parasite from the Plasmodium family. This disease can cause a lot of the same symptoms as the common cold, making it difficult to detect. This same year, Oswaldo Cruz, head of Manguinhos Institute, was researching yellow fever in Rio. Yellow fever is a virus transmitted by female mosquitoes. It is named yellow fever from the jaundice it causes due to impaired liver function in the human body. Chagas spent time after graduation working in Santos, São Paulo, where malaria was becoming an epidemic in the town. Here, he invented pyrithium, an insecticide used to prevent malaria by killing mosquitoes. This proved to be successful. This insecticide comes from chemical compounds from a chrysanthemum daisy. This daisy contains known chemicals that harm and kill insects. After this invention, he started researching and working with Oswaldo Cruz at the Oswaldo Cruz Institute in Rio de Janeiro in 1908. In 1909, Chagas was sent to La Science to investigate a malaria epidemic there. This is where he first discovered American trypanosomiasis, also known as Chagas disease. He spent 2 years here studying malaria, and in these 2 years, he discovered the disease by testing the blood of a 3-year-old girl and discovering a new type of trypanosome in her blood. He discovered that this trypanosome was present in the intestines of the triadenine host bug. He inspected this bug, also known as the Barbero bug or the kissing bug, while researching and proved that this bug is the source of the disease. He named this new parasite T. cruzi after his friend and colleague Oswaldo Cruz. Chagas was also the first to discover pneumocystis pneumonia, caused by the bacteria P. gyrovici. This condition is linked to pneumonia in AIDS patients due to decreased immune function. This is a type of fungal pneumonia. Chagas' contribution to bacteriology, as well as his discovery, led to improved sanitation processes in Brazil, which are still in effect today. In 1920, a National Department of Public Health was established with Chagas as its first director in Brazil. This department is still in effect today. Through his research, Chagas was able to determine diseases that are specific to the tropical regions, such as malaria, Chagas, and yellow fever. His exploration, along with the help of Cruz, promoted further exploration and gave more detailed information regarding the study of vector-borne diseases for future scientists to build off of. Yellow fever and malaria were both epidemics during this time, though not much was known about either of them. Thanks to Chagas, we now know more about host-parasite relationships, and the foundation of our knowledge today regarding malaria and Chagas disease are largely due to the work of Chagas and Cruz. Chagas also raised awareness about health and conditions in rural areas and how these conditions differ from the poverty-stricken areas, putting these poverty-stricken areas at higher risk for diseases. He helped to institutionalize Brazilian medicine and made it what it is today. Ronald Ross and Walter Reed also studied vector-borne diseases, tropical medicine, and protozoology, which further advanced a rather unknown field in their time. Ronald Ross studied malaria and tropical medicine, while Walter Reed specifically studied yellow fever and the mosquito species that carried it. Although all of these scientists were studying diseases spread by insects, these diseases differ greatly in their symptoms, disease processes, and modes of transmission. Each of these scientists discovered information that is vital to the foundation of new discoveries concerning these diseases in present day. Even though these discoveries were made around 18 to 1900, they gave us valuable information which led to our ability to treat, diagnose, and detect these vector-borne diseases today and prevent future epidemics that mimic those of the past.
Mosquitoes have caused problems in the world for thousands of years, but a little over 100 years ago, a man named Walter Reed made a discovery about mosquitoes that would be one of the most important contributions in the field of medicine and human history. Before I explain his discovery and why it was so significant, I need to tell you about Reed himself and how he ended up working with mosquitoes. Walter Reed was born on September 13th in 1851 in Belroy, Virginia. He was the youngest of 5 children, and in 1866, Reed and his family moved to Charlottesville. It was there that Reed set out to study classics at the University of Virginia, but he soon decided he would rather pursue medicine. In only 9 short months, Walter Reed became a doctor of medicine at the age of 17 in 1869 and is still the youngest student to receive the degree at the University School of Medicine. 1 year later, in 1870, Reed attained yet another degree at Bellevue Hospital Medical College in New York. It was at this time that Reed decided to pursue a military career for security purposes. Once in the Army, he successfully passed the exam to become first lieutenant in the Medical Corps in 1875. Soon after, in 1876, Reed married Emily Lawrence and relocated annually for the next 18 years. He did not accomplish much during this time, but in 1889, Reed finally settled down a bit and was appointed attending examiner and surgeon for recruits in Baltimore. After attaining this position, Reed was granted permission to work at Johns Hopkins Hospital. This is where he began to take courses in bacteriology and pathology. A few years later, in 1893, he was assigned professor of bacteriology and microscopy at the Army Medical School. Skipping ahead to 1898, Reed was appointed chairman of a committee that was responsible for investigating typhoid fever during the Spanish-American War. After concluding his typhoid fever investigation, he immediately became involved with the study of yellow fever. Also, more American soldiers died from yellow fever, malaria, and other diseases during the Spanish-American War than from combat. As yellow fever devoured Cubans and Americans, Army Surgeon General George M. Sternberg appointed a commission in 1900 to investigate yellow fever. This is what Walter Reed is known for. The name of the commission was called the Reed Commission for Major Reed, even though there were 3 other specialists in the group: James Carroll, Aristides Agramonte, and Jesse W. Blazer. But before I go any further about Reed, here's some information about yellow fever so you all can know what Reed was dealing with. Yellow fever is a viral infection that is transmitted to humans by mosquitoes. Some of the acute symptoms include fever, obviously, headache, nausea, and dizziness. Some of the more severe symptoms include jaundice, bleeding from the eyes, nose, and mouth, kidney failure, and neural dysfunction. In the 1800s, most people believed that this infection was spread by fomites, and they held this belief as late as 1898. Others, like Cuban physician and epidemiologist Carlos Finlay, thought that yellow fever was transmitted by insects. This was an interesting theory, but he could not prove it. After G. Santarelli, an Italian bacteriologist, isolated Bacillus icteroides from yellow fever patients in 1896, the U.S. Army appointed Reed to investigate Santarelli's bacillus. Okay, now back to Reed. In February of 1900, Reed submitted a complete report which showed that Santarelli's bacillus was the second invader in yellow fever. A few months later, Reed's commission was investigating an outbreak of what was thought to be malaria in Havana, Cuba, Cuba's capital, but Reed found that the disease was actually yellow fever. It was during this time that he made the discovery that yellow fever is not transmitted from person to person, but by insects, specifically Aedes aegypti mosquitoes. You might be asking yourself, how did he come to this conclusion? While he was investigating the yellow fever outbreak, he found that 1 prisoner in the cell in the barracks had yellow fever, but the 8 others that were there remained unaffected. The main work of Reed and his commission would then be to prove or disprove the agency of an insect intermediate host. In order to confirm this, Reed had soldiers volunteer to wear clothing that belonged to people who were infected with yellow fever. As expected, the volunteers did not contract yellow fever. After ruling out person-to-person transmission, Reed tested the mosquito theory in Washington by hatching eggs from Finlay's collection, having them feed on the blood of yellow fever victims, and biting several healthy individuals. His theory was then proven to be correct. When Reed returned to Cuba, he set up Camp Laser, an experimental camp in the jungle, to continue his investigation of yellow fever. Reed and his commission, by demonstration, showed that mosquitoes are able to be vectors of the yellow fever virus if they feed on a person within 3 days of infection. After an incubation period, the mosquitoes could successfully pass the virus on to humans. In February of 1901, under Major William C. Gorgas, U.S. military engineers took action in Cuba on the basis of Reed's findings, and Havana was freed from yellow fever within 3 months. When Reed returned to Washington in February of 1901, he continued his duties as a teacher but died after an appendicitis operation 1 year later, on November 22, 1902. Even though Reed passed away, his research and his findings still apply to the world we live in today. Society still deals with mosquitoes and a number of viruses they are able to carry, but thankfully, Reed's research provided a basis for how viruses were transmitted then and now as well. Walter Reed was able to discover how yellow fever was transmitted, helping to eradicate it. For this reason, Walter Reed and his contributions hold a special place in both human and medical history.
Sir Ronald Ross was born May 13, 1857, to Sir C.C.G. Ross, a general in the Indian Army, and his wife Matilda. Though he had no previous interest in medicine, he followed the instruction of his father and began studying medicine at St. Bartholomew's Hospital Medical College in London at age 17. While studying to gain his licentiate of the Society of Apothecaries, he worked on a transatlantic steamship as a surgeon and then entered the Indian Medical Service in 1881. He began developing his scientific interest and took a course in bacteriology in hopes that he would gain his diploma in public health from the Royal Colleges of Physicians and Surgeons in England. In 1894, Ross started learning about malaria and, more specifically, what a man named Sir Patrick Manson, now known as the father of tropical medicine, had hypothesized about malaria being caused by parasites, and how those parasites were also able to infect mosquitoes. He began talking to Manson as well as a man named Charles Alphonse Laveran on a regular basis in 1895, trying to figure out how exactly mosquitoes and malaria correlate. His progress was brought to a halt when the Indian Medical Service relocated him from Madras to a malaria-free environment in Rajputana. He threatened to resign from his position until the government put him on a special duty for an entire year to specifically investigate the malaria infection. He tried, time and time again, to test his findings that mosquitoes transmitted malaria by taking Culex mosquitoes that had fed on malaria patients and having them bite new, uninfected humans. This didn't seem to work. In a letter to his wife, he wrote, "I have failed in finding parasites in mosquitoes fed on malaria patients, but perhaps I am not using the proper kind of mosquitoes." Then, on August 20th of 1897, now known as World Mosquito Day, Ross made his first huge discovery while dissecting the stomach of an Anopheles mosquito that had been feeding on a malaria patient for 4 days. In a blood smear of the mosquito, he saw 2 parasites right next to each other and went on to prove that transmission only occurs in Anopheles mosquitoes. A year later, he was able to demonstrate that mosquitoes served as an intermediate host for bird malaria. After mosquitoes fed off infected birds, the parasites would enter their bloodstream and migrate towards the insect's salivary glands, allowing the mosquito to infect other birds. Although bird malaria and human malaria are different, This still explains the life cycle of the Plasmodium parasite in Anopheles mosquitoes and how transmission occurs. In 1899, Ross resigned from the Indian Medical Service and returned to England. He became a professor at Liverpool's School of Tropical Medicine as well as a chairman in tropical sanitation at Liverpool University, where he was to investigate and develop anti-malaria schemes in West Africa. In 1902, he received the Nobel Prize for Medicine for his work on malaria, by which he has shown how it enters the organism and thereby has laid the foundation for successful research on this disease and methods of combating it. Without Ross's painstaking research on malaria, we wouldn't fully understand the origins or the transmission of malaria. We wouldn't have specifically targeted anti-malarial treatments, and we wouldn't understand the importance of getting treated before we go places where Anopheles mosquitoes are present. It's fair to say that Ronald Ross saved countless lives through his discovery of the transmissions of malaria and will continue to save lives because of his landmark discovery in 1897.